Have you ever imagined what could happen if you get a paper cut and there is nothing in your system to control the bleeding? You will definitely die of exsanguination. Right, so you have very important formed elements in your blood called the platelets. Platelets originate from megakaryocytes, which are the largest progenitor cells of the bone marrow, and themselves originate from common myeloid progenitor. Right, so you can see here this is a multipotent hematopoietic stem cell, right, giving common myeloid and common lymphoid. Right, so you follow the common myeloid. Here you will see myeloblasts, mast cells, erythrocytes, megakaryocytes, and then from megakaryocytes, that's where you get platelets, also known as thrombocytes. The lifespan of platelets is 8 to 10 days, and they are about 150,000 to 400,000 per cubic millimeter. The spleen stores about a third of the body's platelets, and these are not included in the platelet count. Abnormal platelet count. Right, so if the platelets are more than 400,000 per cubic millimeter, it's called thrombocytosis. If they are less than 150,000 per cubic millimeters, it's called thrombocytopenia. Right, so the platelets contain two types of granules, the alpha granules and dense granules. The alpha granules contain proteins of high molecular weight, including von Willebrand factor, factor V, and fibrinogen. Conversely, the dense granules contain low molecular weight molecules such as ATP, ADP, serotonin, and calcium ions. Platelets have abundant surface receptors, which are classified into agonists and adhesion receptors. Agonist receptors recognize stimulatory molecules, and examples include collagen, thrombin, and ADP, among others. The adhesion receptors promote the adhesion of platelets to other platelets, the blood vessels, or leukocytes, depending on the particular receptor stimulated. Examples of adhesions include glycoprotein 2B3A receptor, which is targeted by antiplatelet drugs such as tirafiban. Right, we will talk about these later. The key role of platelets is their participation in hemostasis through formation of blood clots at the site of bleeding. Right, so what the hell is hemostasis? Right, so if you break this word, hem for blood, stasis, stopping, right? So stopping bleeding. So hemostasis is a physiological process by which a bleeding stops. Its final result is a thrombus or a blood clot, which consists of red blood cells and fibrin strands. Right, so there are two kinds of hemostasis: primary hemostasis and secondary hemostasis. In this video, we are going to focus on primary hemostasis. Right, so this primary hemostasis has two stages as well: vascular hemostasis, which is a transient vasoconstriction, and von Willebrand factor activation following endothelial injury. And second, we have platelet hemostasis. This is adhesion, activation, and aggregation of platelets, which will result in the formation of a platelet plug, also known as white thrombus. Then secondary hemostasis, this is activation of coagulation cascade, which result in the formation of a fibrin clot, thus a red clot. Right, so in primary hemostasis, you have a white clot, only platelets. Secondary hemostasis, this time you have a red thrombus or red clot. In this case, there is fibrin. Right, so let's focus on primary hemostasis. What is it again? This is a process involved in the formation of a platelet plug, white thrombus, following endothelial injury. Vascular hemostasis. Right, so endothelial injury will result in neural stimulation reflexes and release of endothelin. This will lead to transient vasoconstriction, which will lead to reduced blood flow and platelet accumulation at the vessel walls, right, at the site of injury. Right, so here you can see the structure of a blood vessel, right? So inside here you can see this one cell layer. This one is called endothelium, right? And basement membrane and arterial smooth muscle, right? It's just a structure, but here is the area of injury, right? So the after release of endothelin, this will stimulate vasoconstriction, which will reduce blood flow, and the platelets will come at this site. The exposure of subendothelial collagen will lead to circulating von Willebrand factor binding to the exposed collagen. Right, what is von Willebrand factor? This is a plasma protein that is synthesized by and stored in endothelial cells, thus in Weibel palade bodies and in platelets in the alpha granules, right? So they are produced by endothelial cells and by the platelets. Von Willebrand factor mediates platelet adhesion and aggregation. It also binds factor 8, thereby preventing its degradation. Platelet hemostasis. Right, so there are different stages. The first stage is platelet adhesion, right? In this case, the platelets bind to von Willebrand factor via glycoprotein 1b receptor at the endothelial injury site. Ristocetin, right? Ristocetin, you will see this in MCQs. It normally activates von Willebrand factor to bind to the glycoprotein 1b. Second stage is platelet activation. After binding to the von Willebrand factor, the platelet will change their shape and release mediators that lead to activation of more platelets. This is a positive feedback. So these mediators include adenosine diphosphate, that's ADP, which promote adhesion of platelets to endothelium. Thromboxane A2 activates additional platelet and promote vasoconstriction. Calcium. Calcium is required for secondary hemostasis. It's factor 4. I will tell you more about it in the next video. Platelet activating factor. Platelet activating factor, right? So this is a phospholipid mediator that is produced by platelets and inflammatory cells, for example, neutrophils, monocytes, macrophages, and is involved in platelet aggregation and activation of local inflammatory response. Platelet aggregation. This is mediated by glycoprotein 2b3a receptor and fibrinogen. This will lead to formation of a white thrombus composed of platelet and fibrinogen. A white thrombus is transient, unstable, and is easily dislodged, right? So it will be stabilized by secondary hemostasis. Right, so this is just a conclusion of primary hemostasis. Right, so this in purple is von Willebrand factor, and these are platelets. So between von Willebrand factor and platelets, you have glycoprotein 1b. Right, so between platelets themselves, they are glycoprotein 2b3a, right? You can see them, and in the midst, you have fibrinogen, right? So these are the receptors. Right, so we'll talk more about this pathway in the upcoming videos. Stay tuned.